So to really So to really understand how vaccines operate, it's important to first talk about how we respond to natural infection. As we are currently in an ongoing viral pandemic, I think it's best to discuss what happens when you're infected by a virus. When you're infected by a virus, two parts of your immune system activate, both your innate defense system and eventually your adaptive system. What your innate system is incredibly useful for is pattern recognition. What it can do, and it say, oh, this feels like a pathogen, this feels like a virus. I need to tell somebody. And in part of telling somebody, it performs a number of essential functions. These functions help fend off further infection. They include activating neighboring cells to further recruit more help, directly destroying the invading organism, and eventually starting repair mechanisms for the process of healing. The adaptive branch, though, is what really gives us our immune memory and forms the basis of successful vaccines. During a viral infection, two separate adaptive immune response cell population, two separate branches are activated, B cells and T cells. B cells are your antibody producing cells. Initially, when activated, they produce an antibody IgM. This antibody is relatively low affinity and sometimes can be non-specific to the infecting virus. But as the infection progresses through time, B cells kind of wise up to the infecting pathogen. They start to produce a more higher affinity antibody known as IgG. Now, you may know about these IgG antibodies because we've been talking about them a lot with COVID, and we often measure these to look to see if you've had a distant COVID infection. Additionally, B cells can produce two specialized types of antibodies, IgA, which is more specialized to your mucosal surfaces, your lungs, and your gut, in addition to IgE, which plays a specific role in the allergic response. The other population is T cells. T cells, when they encounter a virus, primarily differentiate into CD4 positive and CD8 positive T cell lineages. The CD4 lineage is referred to colloquially as T helper cells. You may know this name because CD4 cells are the target of HIV infection. CD4 positive T cells are the target of HIV infection. What these cells do when you have an immune response is they assist in creating higher affinity antibody responses as well as coordinating the overall immune response. CD8 cells are referred to as killer T cells. They include two separate populations of cytotoxic T cells, which are important for killing intracellular organisms, as well as cancerous or virally infected cells. Another branch is CD8 positive suppressor T cells, which really are important to restrain certain types of immune response. So what is the goal of a vaccine? Well, the goal of the vaccine is to elicit an immune response that is protective. What this means is that when you come into contact with a pathogen that you're trying to protect against, you don't get clinically significant disease. This doesn't necessarily mean that you don't acquire the pathogen, but a good example of that could be the two different polio vaccines, one of which still allows polio to replicate, one of which provides what we call sterilizing immunity. But really the goal of a vaccine is to prevent that clinical outcome. Now, how do we do this? How do we create different vaccines to elicit a protective immune response? Well, there are 5 main formats that we can cover. All these different formats do is they provide a different avenue to tell your immune system to watch out for this invader in the future. So again, let's walk through these formats using the example of a virus. The first of the 5 formats is the live attenuated or weakened virus. And basically what this does is it takes a virus, it weakens it through some kind of mechanism. Sometimes we manipulate the genome, sometimes we serially passage that virus. And what we produce is still an active virus, something that can infect you, but it only infects for a few life cycles, so only 3 or 4 different life cycles, and then is eliminated by its own means or by your body's immune system. A good example of a successful vaccine of a live attenuated virus would be the MMR vaccine. Another version of vaccine is called a killed vaccine. What we mean by that is we go into the laboratory and we take virus, and then we subsequently UV radiate it or chemically fix it, we do something to render the virus unable to infect a host. We then take a lot of that virus and inject it into a host to elicit a good immune response. An example of this would be the killed influenza vaccine. Method number 3 and method number 4, subunit and toxoid, are very similar. A subunit vaccine takes advantage of scientific advancements to artificially produce a known immunogenic viral protein. We then take a protein that we know if we can get a good immune response to, it's protective, and we give it to a host and subsequently mount that protective immune response. But a good example of this would be hepatitis B. Similar to this is a toxoid vaccine, more relevant for bacteria. We take a purified toxin of the bacteria and then subsequently give it to a host and allow you to create a good immune response against that bacteria. A good example would be diphtheria. And of course, the fifth and final format, the most famous, the most in vogue, is our new mRNA vaccines. This takes technology that is somewhat similar to how we artificially produce proteins in the laboratory, and then applies it to deliver mRNA into human cells to produce immunogenic proteins. The wonderful thing about this platform is that it's highly adaptable, meaning that instead of producing proteins or virus and growing up large stocks of immunogenic material, instead we can produce mRNA in record time and subsequently respond to pandemics faster. Now we know the two branches of the adaptive immune system, and we additionally know generally what a vaccine is trying to achieve. But the question is, that's how a vaccine is trying to achieve. What's the avenue of which it actually does this? Well, you can guess by the big red box in the slide, the B cell plays a major role in how we think vaccines provide protection. Now, through multiple clinical trials across the globe with differing pathogens, a major lesson of vaccinology is the B cell response that produces a good IgG antibody response. It's the primary method through which a lot of current vaccines provide protection. But what vaccines am I particularly talking about? Well, let me list a few. Hepatitis A, a major cause of acute inflammation of the liver with significant abdominal pain. We created a vaccine against this virus and have seen a 76% decrease since the vaccine was introduced with cases of Hep A. Hep B we've already talked about. This can cause a chronic illness leading to liver cirrhosis and subsequently perhaps requiring a transplant. Hepatitis B killed around 820,000 people globally in 2019. We have a very effective vaccine. We've talked about mumps, measles, and rubella, the MMR vaccine. These were mostly pediatric illnesses, but now we've created a vaccine, and through our good antibody response, we've eliminated a lot of these as significant causes of pediatric morbidity and mortality. Influenza, one of the leading causes of ID-related geriatric hospital admissions. We create a new vaccine every year, which we know has varying efficacy, but still antibodies play a major role in how this vaccine functions. Rotavirus, a brutal pediatric diarrheal illness, now able to be prevented through a vaccine and antibodies. Smallpox, the best story you've ever heard, eliminated except for a few vials across the earth. JV, the number one cause of paralytic viral illness in Asia, now has a vaccine, 99.9% effective. And I'm going to throw up here also rabies. We're developing multiple new vaccines to rabies that have shown that antibodies play a major role in how they can protect against human hosts and also provide protection to animal models. This is a lot of different pathogens, but I'm also excluding a few because there are some pathogens which we identified that antibodies aren't the primary way to provide protection. And a lot of these unsolved pathogens require a good T cell response. One of the major problems across the globe is tuberculosis. Well, TB is an intracellular organism. It's hiding within cells. It's able to invade your lungs and then hide within different cells within the lungs and form these large granulomas. And it really requires a good T cell response to be able to clear this bacteria. Another good example is chlamydia. Chlamydia spends the majority of its life cycle within the actual cell, only lysing out for a little bit of time before subsequently re-entering another cell. As antibodies often act extracellularly, it's very difficult for them to prevent further infection when chlamydia basically pops out and then is able to go on and infect another cell. And so when we're looking for the next frontier of vaccines, we're really looking to develop, as one arm of vaccinology, are good T-cell vaccines. And one of the major questions Dr. Darville will address is how do we make vaccines that produce a good, robust T-cell response that can help clear some of these intracellular organisms, with the main focus being on chlamydia. And we always end these student talks with more of a take-home message, something that you can go to your physician with. And basically, the big take-home message is go and get vaccinated. The Carolina provides a good database of vaccines you've already received, but every year you're eligible for a different flu vaccine that may have varying efficacy, but can provide some protection and prevent severe illness. Additionally, if you're a little bit older in age, varicella is a wonderful vaccine, as well as these two vaccines here that are able to prevent a lot of pneumonia. So if you think you're eligible for these vaccines based on age, ask your doctor, follow up with them, and try to get your vaccines at your next primary care appointment. And with that, I will take any questions.
All right, Cameron, thank you very much for that wonderful talk. And to the audience, please feel free to address your questions to Cam in the Q&A chat box that you can see at the bottom of your screen. Yeah, thanks.
I guess while we wait for people to get typing, I have a question for Cam. Does T-cell immunity play a role for virus immune response in vaccines?
Yeah, T-cell immunity does play a role in the viral response for vaccines, because a lot of you need T-cell help to produce really a long-lasting immunity. But as far as actually neutralizing viruses before they're able to infect your, like being able to stop them before they can infect a host, a lot of that is performed by neutralizing antibodies. But saying that T cells are irrelevant is wrong. T cells are very, very important to give you a good, robust response.
All right, we do have a question in the chat. Jackson, if I can find you, I'm going to unmute you to let you ask that question to Cam. You should be able to unmute yourself now. There you go.
Yes, can you hear me?
Yeah, I can.
Okay, cool. My question was, what causes such a strong response in some individuals for the COVID-19 vaccine? Like, for example, for me, after my doses and booster, I was down for a few days. And obviously glad that it's protecting me from COVID, but I was just curious.
It sounds like you had a really good immune response based upon just saying you've been down for a few days, but I think we're still trying to understand why a lot of folks felt a little bit crummy after getting the vaccine. Just talking to other pediatric infectious folks and other infectious disease folks within UNC, a lot of thoughts are a lot of those severe responses, not meaning severe, but more meaning like, you felt crummy for a few days, happened for folks that had really strong immune systems. It was more in the 20-year-old to 40-year-old range, folks that responded vigorously to the vaccine. So that would be my guess, but there's still more information for us to gather to really understand that.
That's really interesting. Thank you so much.
If there's not another question in the chat, I have one that you could maybe answer as well, Cam. So some vaccines need boosters and some don't. do you have any insight as to why like some vaccines would and some vaccines would not?
We're still understanding this. A lot of immunity will wane with age. So as you get older, you are going to need boosters. The goal really is just to provide that long lasting, significant immune response, something that is protective. And I think the actual mechanism we don't necessarily know quite yet. It seems like a lot of the live vaccines provide a longer duration of response than the killed vaccines. But with that being said, what we do know clinically is that they are required, because when we take your serum and we go and test it in our labs, we can say, oh, you don't have the thing that we know is protected. You often don't have a lot of these antibodies. Take another booster, boost those antibodies up. So let's not take the risk that, we just caught you on a bad day and there's no antibodies circulating. Let's just get you back to where we know is protected.
Thanks, Cam. We have one more question that just came in the chat.
Yeah, let me just go and find you, Sarah. All right, you should be able to unmute yourself now.
Hey, thank you. Yeah, I was just curious. I know with pregnant women, they're not supposed to take the MMR because it's a live vaccine. So I'm just trying to think, what do live vaccines do to fetuses with respect to B and T cell reactions?
I think as far as the MMR vaccine, it's the actual testing on pregnant women that suit a little bit further. But as far as taking a live vaccine, I'm not sure the exact immune response that has to happen that may put a fetus in danger, but you are somewhat immunosuppressed due to some T cell response suppression. So it may not be as effective, but I think we just don't know and we're not willing to take the risk.
Got it. Thank you.
Okay. If there are no more questions for Cam, we can go ahead and take a quick break and come back in, what do you say, Kev, 10 or 15 minutes?
Let's bring it back at 7 o'clock and give
Right.
Dr. Darville as much time as possible to restart.
That sounds great. All right, so we'll see you all back at 7 Eastern time.
Thanks, Kevin. And thanks, Nate, and to all of you guys for planning this mini medical school. I think it's great for us to reach out to the community and share with them some of the knowledge that we have and the research that we do. So I'll go ahead and share my screen. As Kevin said, I've been studying chlamydia for a long time, and my goal is to teach you some about this sexually transmitted infection and how at UNC we're working really hard to try to develop a vaccine against this infection. So I'm going to share with you some information about chlamydia and our chlamydia vaccine initiative at UNC. And what we're trying to determine is what, who, and how to prevent this infection. So first off, chlamydia is not a flower. I've seen this on t-shirts and posters. Although the word is a pretty word, It definitely is not a pretty infection or pretty disease. It causes some bad diseases. And so if you take home anything from this session, just remember it's not a flower and you don't want it. It's the nation's most prevalent sexually transmitted infection. So an outline of my talk is, first I'm going to tell you what is chlamydia, and then what diseases chlamydia causes, and then discuss the question, can we make a preventative vaccine? And if you're going to make a preventative vaccine, as Cam mentioned, you first need to know what immune responses combat the bacteria or the virus that you're trying to prevent. And then can we induce those responses with a vaccine? And then finally, once we get the vaccine, how are we going to test it? How are we going to get it to the clinic? So Cam showed some pictures, and this is actually a fancy, high powered electron microscope picture of chlamydia infecting a cell. And you can see here, there's a small spore-like structure, which is the infectious elementary body form of chlamydia. And it binds to the cell in a myriad of different ways and is taken up by the cell very quickly. And as soon as it gets in, it wraps itself in part of the cell membrane. So it actually hides like it's supposed to be in the cell. And once it gets in there, it converts to a multiplying form called the reticulate body. And you can see, here's a picture of reticulate body 2 hours after uptake. And then it starts multiplying by just division. So it divides and divides, all the while stealing nutrients from the host cell to make progeny or daughter cells or daughter chlamydia. And so once you get about 300 new chlamydia in a cell, it finally exhausts the cell of its nutrients, and it gets so big that it actually can push the nucleus of the cell over to the side. And so here, this picture is 36 hours after infection. And so at this point, the replicating forms then convert back into these spore-like forms for release from the cell so they can go infect other cells or another unsuspecting host. So this is a cartoon of what I just showed you in that microscope picture. You can see here, here's the elementary bodies. They're escaping from this cell. They bind to neighboring cells and enter very quickly. They convert into this replicating form. They divide and divide and divide and steal nutrients from the host. And then finally, once they want to be released, they convert back into the spore-like form, and they basically explode the cell and are released through cell lysis, damaging the cell and the tissue, or they can even balloon off and then be released from the cells. What's really interesting about chlamydia is when you stress it by immune response or perhaps other co-pathogens that may be involved, it can actually become kind of a dormant state where it just hides in the cell and sits there. And then once those stresses are removed, it goes back into its replicating, multiplying form. And so because this organism is so successful in the cell, it makes it hard to get rid of it with our immune response, and it makes it difficult to prevent infection. So this is a picture of chlamydia organisms in these vacuoles or these sacs, and they've all multiplied up. And you can see here's the nucleus, and these are human cervical cells in a dish. And the thing that happens when you get this big bag of chlamydia in your cell is the cell's not happy. And so it sends out signals, and that brings in inflammatory cells. Neutrophils come in, and those actually release toxins and damaging molecules that can actually inhibit the chlamydia, but at the same time, they can affect the health of the tissue. Lymphocytes, which are pictured here, also come in, and those are the cells that we really want to come in to protect us from chlamydia, because they release a protein called interferon gamma, and interferon gamma inhibits chlamydia. So what human diseases does chlamydia cause? Well, we don't think about it much in the United States, but actually in low and middle income countries, Ethiopia, Tanzania, Gambia, one of the most common causes of infectious blindness is chlamydia. And it basically infects the lining of the eye, the conjunctiva, this membrane right here. And that leads to inflammation and scarring. And this happens over and over and over in these villages. And finally, the scarring causes the eyelashes to turn inward and scrape against the cornea. And so by the time these people have sustained multiple infections with chlamydia, they develop blindness, sometimes in their early 20s or a little later. So fortunately, there are control programs in these areas, which are leading to improvement and resolution of these infections. In the United States and higher income countries across the world, the main thing we worry about is what chlamydia does to women. It infects the cervix of young girls and young women, and then it actually ascends to the upper genital tract, and it can affect the uterus and the oviducts, where the ovum or the egg has to travel down to the uterus to become fertilized. And this leads to scarring of the oviduct, which can block the egg. You can see here, this is a patient at surgery where they've taken a picture, and here's the uterus, and you can see the oviducts or the fallopian tubes are all swollen and red and edematous, and there's some purulent, pussy material coming out. So this patient is in a bad way, and they probably have severe lower abdominal pain because of their infection. In the Gambia and Ghana and Ethiopia and areas, the Carter Center has really done a great job of instituting chlamydia trachoma control programs. So you can see all these kids are really happy. Their eyes are not infected with chlamydia. And this is because they basically go into entire villages and give everyone antibiotics multiple times a year. And this has really been helpful to prevent chlamydia in these areas. Here in the United States and in Europe and other areas of the world, chlamydia is one of the most common causes of genital tract infection in men and women. In men, it infects the urethra, and it can cause painful urination, penile discharge, painful ejaculation, and even up to the testicles, it can cause testicular swelling. In women, it loves to live in the cervix, but then it also can ascend, as we talked about, to the upper genital tract. These infections can lead to painful intercourse, vaginal discharge, bleeding between periods, and lower pelvic pain. So in a woman that's infected in their cervix, this can lead to sexual transmission, can lead to vaginal discharge, but it doesn't cause any chronic pathology. But when the organism ascends to the upper genital tract, it can lead to acute pelvic pain, and those patients will present to the emergency room or clinic with pain. But it can also ascend the upper genital tract and produce silent disease, which is a little scary because girls and women are walking around with this infection. This can lead to chronic complications of infertility, chronic pelvic pain, and even ectopic pregnancy. This is a picture of a dye test that's used on a woman who's presented to a clinic because she's struggling to get pregnant. On this side, you can see a normal study. They basically put a catheter up into the uterus and they squirt dye. And when your fallopian tubes are wide open, like they should be, the dye leaks out of the fallopian tubes into the pelvic cavity, which you can see here. This woman who's having fertility problems has blocked tubes. So basically there's dye in the uterus. It gets up to the end of the fallopian tube, but you can see it doesn't spill into the pelvic. These are both blocked. It's as if you took a garden hose and tied a big knot in it. And so what happens is probable to the knot very swift, and that can lead to ectopic pregnancy, where the sperm actually ascends and fertilizes the egg in the oviduct, and the fetus begins growing in this very small structure. And this causes women to present to the emergency room with acute hemorrhage, and they can die from ectopic pregnancy. So the long-term complications in women can be very severe. Chlamydia is the leading cause of involuntary infertility in the world. And my gynecological colleagues tell me of patients all the time who are coming in with chronic pelvic pain after they've sustained this infection. If untreated, around 10% of infected girls will develop acute pain or pelvic inflammatory disease. But the chronic complications occur in about 10% of those. And these are the severe complications of infertility and risk for ectopic pregnancy. When a mother has a baby and she's infected at the time of delivery, even the baby can develop chlamydia infection. It can infect the eyes, as in this newborn. It can infect the nose and lungs and cause them. Fortunately, in developed areas of the world, all the women that are pregnant are tested for chlamydia during pregnancy. And so in our areas. Well, how prevalent is chlamydia? It's probably more prevalent than you would think. In fact, in the southern states, it's quite prevalent, with rates of over 500 per 100,000 population, just with widespread screening. They actually call this the Southern Sexually Transmitted Infection Belt, because southern states are so prone to have higher rates of sexually transmitted infection than other areas of the US. It is the #1 bacterial sexually transmitted infection, as 1 in 10 sexually active adolescent girls test positive for chlamydia. When I was in Arkansas, we did a study in our adolescent clinic, and in that clinic, where girls were coming in for birth control and healthcare, 1 in 5 girls were infected. So it's very common in the South. It's so common because most people don't have acute symptoms. In fact, 90% of infected female teenagers can have infection and have no symptoms, so they don't know they're infected. And boys are similar. Boys have about 70% rate of asymptomatic infection. So if you don't know you're infected, you don't go to the doctor, and you continue to spread infection if you're practicing unprotected sex. So this infection is diagnosed primarily through screening programs when young teenagers or young adults, older adults come into their doctor for their routine health care and are screened for infection. This is when you find it. Well, fortunately for chlamydia, we have antibiotics that are very effective at killing this pathogen. Doxycycline is one, azithromycin is another. Both of these drugs work very well on intracellular pathogens, which chlamydia is one of those. So why not just screen and treat everybody with antibiotics? Why do we need to worry about a vaccine? Well, screening and treatment programs do work to reduce disease, but you can imagine it's very hard to maintain them. They're extremely expensive and logistically difficult to keep people coming in and being screened and then treating them, and then they go back out and get reinfected. So it requires a very high level of sustained effort to do this. And despite screening and treatment programs in the United States, we're seeing these rates of chlamydia continue to rise. You can see these are the rates in women, which are higher than men, likely because women are more often screened. But there's definitely no decrease in sight over the past 20 years. So I've told you what chlamydia is. I've told you what diseases it causes. So now the question is, well, can we make a vaccine to prevent this infection and all the problems, especially the chronic problems that it causes in women? Well, we've asked the question for many years in our laboratory, what immune responses are required for protection from this organism? And one of the reasons is that we ask these questions is that the answers guide vaccine components and vaccine platforms that we want to use in the clinic. So an example, which everybody's learned about SARS, probably lay people know more about vaccine development after the SARS epidemic than we've known for many, many years, which is a wonderful thing despite this pandemic. So for SARS and for other viruses, as Cam mentioned, these viruses have specific proteins on their surface that allow them to attach to human cells and infect. So for these kinds of infections, what you want is an antibody to come in and block that binding from happening. So those vaccines are fairly easy to make. And so if you think about a lot of the vaccines that we use in children, this is the mechanism: flu, hepatitis A, hepatitis B, the pneumococcal vaccine. All of those block infection. Another virus that this strategy is used for is the human papillomavirus that causes cervical infection. This vaccine for HPV drives the induction of antibodies from B cells that block infection of this virus. And it's done a really great job where it's been used to prevent cervical cancer and is now being used in men to prevent cancers of the mouth and oral pharynx as well. So, but if you go back to chlamydia, the problem with chlamydia is that there's not a single protein on the chlamydia cell that allows it to infect a cell. Chlamydia gets in the cell in a myriad of ways, countless ways. It just loves to get in, and it's figured out how to get in. And so there's no antibody that we can make that binds up this elementary body to prevent it from getting in. So we need a different strategy for chlamydia to attack the form once it gets in the cell. So what we've learned over the years is that for chlamydia, what we really need are T cells, CD4 T cells, that make this protein called interferon gamma. And interferon gamma inhibits chlamydial growth by starving it. Chlamydia needs an amino acid called tryptophan to multiply and grow. And tryptophan is provided by the host cell. And interferon gamma induces the metabolism of tryptophan, so that it's no longer present for chlamydia to grow. And this kills off the chlamydia. So for chlamydia, what we need are these cells to stimulate Th1 cells, CD4 T cells that make interferon gamma to inhibit this infection. So in our lab, what we're trying to do now is determine what proteins, what chlamydia proteins or antigens excite or activate these Th1 cells to lead to the production of interferon gamma. Or how are we approaching this? So now we know what immune responses are required for protection. We need Th1 cells making interferon gamma. So in our lab, we are collaborating with others across UNC to find proteins that women who have been exposed to chlamydia recognize and induce T cell interferon gamma response. I was very fortunate to live in Pittsburgh for several years, and there's a huge women's hospital where they deliver about 5,000 babies every year. And so lots of women come to this hospital. And with our gynecological colleagues there, we were able to recruit 247 women into a study to study their T cell responses against chlamydia. These are women who were 18 to 35 years of age. Many of them already had a positive chlamydia screening test, or they came in and said, you know, I hear my boyfriend has chlamydia, I need to be tested. We asked them a lot of questions about their sexual history and their behavioral history, and then we took blood from them, and we also did vaginal and cervical and even a uterine sampling to look at their various sections of their genital tract to see if chlamydia had actually ascended. And then we followed them. And the good thing is about Pittsburgh for a long-term study is that there's lots of bridges and tunnels, and people tend to stay in their neighborhood. So it's easy to get women back into the clinic. So we're able to follow these women and have them rechecked every so often for a year after their enrollment. And what we found is that 65% of the girls that we enrolled were infected with chlamydia when we enrolled them. About half of them were found to have infection and chlamydia up in their upper genital tract. When we followed them for a year, 35% of the women had another positive test during follow-up, but 65% did not. Well, so what we're doing now is we're taking their blood here at UNC, and we're testing them against a whole list of chlamydial proteins that we think might be good antigens. And what we're trying to find out is, do these proteins stimulate their blood cells to make interferon gamma? So these patients have seen chlamydia before. Their T cells recognize chlamydia proteins. And what we're probing for are what proteins are recognized by their T cells and make interferon gamma be produced. So you can see here, if across the rows, you see have patients, and these are different proteins. A lot of these patients are responding to protein C to make interferon gamma. So protein C might be a good protein for our vaccine. So can we induce these protective responses with the vaccines? Well, now we've identified proteins that induce interferon gamma from women who've been exposed. So what platform might we put those proteins in to stimulate Th1 cells with a vaccine? Well, we've heard a lot about the COVID vaccine, and actually, when the COVID vaccine was developed, it was developed to induce antibodies. But what we found out from multiple studies that have been that have happened over the pandemic is that these mRNA vaccines not only induce strong antibody responses, but they also drive really great Th1 cells that make interferon gamma. And that's because of the way the mRNA is packaged in this lipid nanoparticle. If you think about a little kid that hates broccoli, but if you put cheese all over their broccoli, then they're going to eat the broccoli. Well, it's very similar with these mRNA vaccines. Cells don't like to take up mRNA. And in fact, if you just shot mRNA in your body, it would be degraded really quickly. But scientists became really smart when they started putting the mRNA in this lipid nanoparticle, or basically putting cheese all over this mRNA. And when that happens, the cells take it up, they make the protein, and it not only induces antibody, but can also induce T cells. So if you look at the COVID vaccine again, as an example, here's our mRNA covered with cheese. The cell loves it, it takes it up, it makes the spike protein of COVID, and this spike protein gets out of cells, it stimulates antibody production, but can also stimulate T cells. And so we are basically taking this exact same platform and using it for chlamydia. We're fine if antibodies are being made, but what we really want are these Th1 cells. So now we have proteins that we know women recognize and induce interferon gamma. We're putting them in these mRNA vaccines in lipid nanoparticles, and then we're putting them into mice and seeing if they can drive interferon gamma responses, and also can they prevent infection when the mice are challenged with chlamydia. We take the best ones and we actually put them in guinea pigs, because with guinea pigs, we can actually test to see if we can prevent transmission from a male guinea pig to a female guinea pig. So let's say we do all this and we get a vaccine that works great in mice and works great in guinea pigs. Well, then how are we going to take it to the clinic? Well, first you do what we call phase 1, which is a safety and immune response evaluation. And then we do a phase 2, which is to look for actual protection. So for chlamydia, for a phase 1 vaccine trial, you're basically looking for safety. You want to make sure your vaccine is safe in people and that it's going to drive the response that you want to see. So you recruit a bunch of 18 to 25-year-old sexually active adults and assess the safety and the immune response. And you want to look for these interferon gamma producing cells to your vaccine. Then for a phase 2 vaccine trial, you recruit these young adults as well, because chlamydia is much more prevalent in young adults than it is older persons. Because probably after multiple exposures as a teenager, as a young adult, people develop immunity on their own. So you'd basically recruit sexually active young adults, adjust for prior exposure. You could give one group your chlamydia vaccine, the other group a tetanus booster, and then follow them to see who gets infected and who doesn't. Then you would basically do efficacy monitoring. So for men, you can just do a urine test or a urethral swab. Men much prefer peeing in a cup than providing a urethral swab. For women, you can just use self-obtained vaginal swabs and basically check them at intervals and see if they're positive for infection. You would definitely ask them questions about their sexual exposure. And if they had a positive test, you immediately treat them with antibiotics. If they're positive, you can even determine how much chlamydia is there by a test called a PCR. So I've basically told you what chlamydia is, what diseases it causes, and I think we actually can make a preventative vaccine. Now that there's platforms available to us that drive Th1 responses that inhibit chlamydia. And finally, if we get a vaccine, I think it actually would be very testable in young people who are concerned about their reproductive health. And I'll stop there and entertain any questions.
Awesome. Thank you so much. That was a really great talk. I have never heard of lipid nanoparticles as the cheese sauce to broccoli before, but my mom's on the call and she can attest to the efficacy of coating vegetables and cheese for getting people to eat them. So Q&A is open, or feel free to raise your hand in the Zoom menu and we'll get to you as soon as we see it.
In the meantime, Dr. Darville, I have a question.
Sure.
So this is a really, really cool vaccine technology. And I know that chlamydia is not the only intercellular bacterial pathogen that we commonly encounter. Do you think your method is one that would be extrapolatable to, say, tuberculosis or other intercellular pathogens?
For sure, Nate. And actually there are, if you look at clinicaltrials.gov, there are mRNA vaccines that are entering into clinical trials for TB and for malaria. And although the mRNA and a lipid nanoparticle is a great one, there are actually others that I didn't mention, but they're the adenoviral vectors, and so the AstraZeneca vaccine that was given widely in Europe for COVID is actually a DNA vaccine, and it also drives T cell responses as well. So fortunately, over the past, I'd say 10 to 15 years, we've suddenly developed platforms that aren't focused on directly on antibody, but have expanded to induce T cell responses. So really, for the first time in my career, do we have platforms available that can drive a response that can perhaps prevent organisms like chlamydia and TB, those that live their life mostly within the cell.
Awesome, thank you.
That's a question from me too. What makes guinea pigs a good system for chlamydia vaccines instead of like a normal lab mouse?
That's a great question, Kevin. And what's interesting about chlamydia is it spares no hosts. It infects every animal that you can think of. So like 95% of the pigs in North Carolina are positive for chlamydia or have antibodies to chlamydia. It infects koala bears. So it's a huge problem in Australia because it causes infertility of the koalas. So you could actually use koalas as your model. And we actually have done that with our friends at the Queensland University in Brisbane. You can do studies on pigs, big pigs, which we are doing with collaborators at NC State. Or you can use mini pigs. Guinea pigs are good because they are very much like humans. Their reproductive cycle is 18 days instead of 28, but they can be mated, and you can actually, so you can infect a male guinea pig and then put them in a cage with a female guinea pig. And you have to let them like each other for a little bit first, but then you can actually determine transmission. And so it's a very unique model that allows us to vaccinate, let's say, a female and see if we can prevent transmission from a male. Guinea pigs even spread it to their newborns, just like humans. So if you have newborn infected female guinea pig and she has baby guinea pigs, they get infected in their eyes, they get pneumonia. And the pathology that they develop in their genital tract is very similar to what women sustain. This is also true for mice. So we're very fortunate, those of us who study chlamydia, that there are really great animal models that mirror human disease.
That's really great. Thank you, Toni. for the audience out there, I know STIs are somewhat of a sensitive topic, so if you don't want to read your question out and you have one, I'm more than happy to read out your question for you, and that way you don't have to ask a question you don't feel comfortable asking. If not, I have another question. We did talk a little bit about antibiotics as well. And I was wondering if you had any insight onto whether or not chlamydia is becoming antibiotic resistant like many other pathogens that we know of.
Fortunately, the answer is no. So chlamydia has such a small genome that if it mutates its genes very much, it makes it very, vulnerable. So it has a real hard time mutating genes to make it resistant to antibiotics. So that's the one fortunate thing about chlamydia, is that you can induce resistance in vitro if you try really hard. But there's never been cases of resistant chlamydia identified, or they're extremely, extremely rare. So the antibiotics work very well. The problem is, it's just as soon as you get rid of it, if after about 3 months, your immunity wanes after natural infection, and then you get reinfected. So that's the kind of the vicious cycle of infection, treatment, reinfection, treatment.
Okay.
Which is why we need a vaccine.
Yeah. So we've had a few questions pop up in the past moment. The first one says, should sexually active young people be tested for STDs? What if they only have one partner?
I definitely think sexually active young people need to be tested for STDs. If you use a condom, practice safe sex, then you're protecting yourself from sexually transmitted infections. But you may have one partner, but your one partner may not always just have you as their partner. And with chlamydia, it can last for months. So, you may be in a monogamous relationship, but your partner, if they were ever in a relationship months ago with someone else, they could have chlamydia and not know it. And so, that's the issue. So, and it is very highly prevalent, I think, for that reason, because you can have more chronic infections that can last for weeks to months. There's been documented infections in women that have lasted up to a year. And so it's kind of like a stealth organism. It can hang with you for a long time without causing many symptoms, and then yet spread to another individual.
Thank you. And we have a couple more questions. The one, if a pregnant mother tests positive for chlamydia, can she take drugs to treat chlamydia so that the newborn is also cured?
Yes, they basically give the pregnant moms azithromycin, and that's a safe, or amoxicillin. It can even work in pregnant women. And so both of those drugs are useful in pregnant women, and then rescreen the pregnant mom to make sure that the infection is gone before she delivers the baby. So very effective mechanisms to prevent transmission to newborns.
Okay, and so we've also received 2 more questions in the chat that I think we can kind of roll into one. The first question is, in the same vein as antibiotic resistance, has the widespread usage of antibiotics in the communities mentioned earlier, has that led to antibiotic resistance in other illnesses there? And then sort of on another public health vein, if a vaccine did exist, would you give that to both men and women, or both? Men, women, or both?
Okay. So for the first question about antibiotic resistance, so actually what was discovered in these low and middle income countries where trachoma, the eye disease, is endemic, because there was some concern about, developing resistance in other bacterial pathogens. But actually what they found was that these mass treatment programs actually not only prevented trachoma, but lowered mortality, infant mortality in a lot of these areas, because it prevented infection with other bacterial pathogens. Now, these are in small endemic villages and communities. I'm not proposing that, the entire United States should get a dose of azithromycin to clear up chlamydia. So in that sense, it would be a bad thing. And in fact, there's another sexually transmitted organism called Mycoplasma, which can cause chronic penile pain in males. And it causes similar symptoms to chlamydia in females, but not quite as severe. It does get resistant really quick when it's exposed to antibiotics. And so what we're seeing because of treatment of chlamydia is that this mycoplasma organism is developing resistance and becoming much more difficult to treat.
So I guess
the second one was, would you vaccinate both sexes, both genders? Yes. What we've found through mouse studies is that if you vaccinate one, males or females, then it's not nearly as good as vaccinating both. And so if you vaccinate a male and a female, then you can actually prevent transmission. But if you just vaccinate one, it's not enough. And so I think definitely the vaccine would be much more effective if we vaccinate both males and females. And in fact, some studies I did years ago in Arkansas with Roger Rank and male guinea pigs showed that the vaccine, we did some vaccine testing in male guinea pigs, and they actually sustained a better response in females, were more resistant to challenge infections than females, potentially due to just the flushing action of urine, through the urethra. It helps to clear infection when you've got antibodies and immune cells present. But definitely, if you lower the burden in an infected male so even if you can't totally get rid of the infection, if you can just lower how much chlamydia they have, then that can actually lower the rate of transmission. So if you can dampen the infectious burden in both sexes, then you can definitely drive what we call herd immunity, which is prevention of infectious spread.
Awesome. I'm going to quickly skip out of order in the questions for a second to stay on vaccines. Do you expect this is going to be a vaccine for younger people or one that will be used for adults of all ages? Or I guess another way to put that is, what's the optimal age for giving this vaccine? Is it going to be similar to HPV?
Well, for sure, because, the goal will be to prevent the long-term complications in young women. So you want to prevent those by administering it to young girls and women. But I will say that sexual practices are changing, and there's actually been an upswing of chlamydia among nursing homes, and elderly facilities have been described where, we kind of thought it's the young adults and then no more. But it can definitely affect older adults as well. But really, the population of individuals who are most affected by this are women of childbearing age. What happens is teenagers and young women get it and they get over it, and then they go on about their business. And then in their late 20s, early 30s, they're wanting to have a family. And all of a sudden they can't get pregnant. And they're like, what's going on? And they find out that their tubes are blocked. Now, if you have a good financial situation, you can use assisted reproductive technologies and do in vitro, basically have fertilized embryo implanted in your uterus. But if you don't have lots of money, then those technologies are difficult to access. And so, it can be difficult. And in my mind, anyone that wants to have a child should have the right to have a child. So, it's difficult because women who are oftentimes most affected by this disease are the ones that don't have the financial resources to seek in vitro fertilization and assisted reproductive technologies later in life.
Awesome. Thank you. So we've got quite a few questions coming in now. The one that's been hanging out longest is going back to chlamydia conjunctivitis. Why does it affect eye cells? Are there similar characteristics between the eye cells and the genital cells that makes them particularly susceptible?
It's interesting. There's strains of chlamydia that love the eye and they rarely go to the genital tract. And there's strains of chlamydia that love the genital tract. And they are different. And they're different in their ability to make tryptophan. And it just shows you how tryptophan is so important for chlamydia. The eye strains can make their own. The genital tract strains can't. But the genital tract is loaded with other microflora that can help provide tryptophan. So that may have something to do with the different sites that these organisms infect. The organisms that infect the eye are in villages where water is difficult, sanitation is poor. So there's, latrines out back, a lot of flies and insects. And so it tends to be spread from person to person in these manners. But they definitely are different strains than those that infect the genital tract.
Great. Another question. Why is chlamydia so prevalent in the South and many animals besides humans? Do we know of its origin?
That's really interesting. It was discovered years ago when folks they had symptoms like gonorrhea, but they couldn't find any gonorrhea. And so the doctors were like, well, there's no gonorrhea. What's doing this? And then they saw these, under a microscope, these tiny little organisms in cells. And that's the way they discovered it. So it was first discovered in humans. But it definitely, infects duck, birds, fowls. Chlamydia psittaci can, if you have a little pet parakeet and it's, you kiss it on the beak, you can get chlamydia in your lungs. It's a different strain, but it make you really, really sick. So they think that it evolved actually from almost what they call archaea bacteria. So it's such a small form. And then it evolved mechanisms to basically change into this replicating form within a cell. But yeah, it's been around for, as long as there's animals have existed. It even infects snakes, reptiles.
It sounds like it's pretty universal. So we've got a few more questions that we'll try to get through. The first one, once a chlamydia vaccine has been established, do you imagine there'll be similar hesitancy or resistance among parents agreeing to vaccinate their children for an STI, particularly like we've seen in the HPV vaccine?
Oh, I'm sure. Well, if you think about the resistance and hesitancy we've had among our population to protect themselves from, this pandemic. And so, for a sexually transmitted infection, I can imagine there will be definitely some hesitancy. On the other side of the coin, I think there's a lot of young people who are advocating for themselves and for their reproductive health. And so that would be the population I would think that would welcome such a vaccine. And not every girl that develops chlamydia develops these long-term complications. So unlike HPV, where, you basically really have to get them before their sexual debut. If they get HPV, it sticks with them forever, and then they've got this imposed risk of cervical cancer. With chlamydia, it may take multiple infections before someone's tubes are, scarred down and destroyed. So if we can get them early on when they're, in those sexually active years, I think that would be a very good thing.
All right. Okay. Second to last question. Is there any evidence that suggests that a foreskin on a penis increases the transmissibility of chlamydia?
That's really interesting. And actually, we have data that says yes, and there's other studies that say yes. One of the risk factors for repeated infection in our Pittsburgh cohort was having sex with an uncircumcised male. And this has been shown for other STIs also.
Interesting. On a similar note, is chlamydia transmissible from men that have sex with men or women that have sex with women at a similar rate, or not? Do you know?
Men who have sex with men, definitely. Women who have sex with women, unlikely. It can't live. It basically has to have a lot of, healthy, it basically has to have a lot of healthy cells around and go from a person to a person. Women who are only have lesbian sex have really low rates of chlamydia, but men who have sex with men, it can definitely infect the rectum. It can infect the pharynx, the oral pharynx. So you can get it through, oral genital sex. But for women to women, it's more difficult. But yeah, there's a strain that that's more virulent that infects men who have sex with men. And it can actually spread to their lymph nodes, drain their genital tract region, and cause severe swelling in those lymph nodes. And in men who have rectal sex, it can cause rectal infection, inflammation of the rectum, what we call proctitis, that can be quite severe.
Awesome. Thank you very much. And for our final question of the evening, do you expect that the vaccine we are developing will also be used to help battle against Trachomatis trichiasis?
Yes. Yeah. I think definitely the proteins that we're finding that people recognize that induce gamma interferon are conserved in the strains that cause genital tract infection, as well as those that cause trachoma. And so I think that would be an added benefit of developing a vaccine, so that it could not only be used to prevent sexually transmitted infections in developed areas of the world, but also trachoma in low and middle income countries.
Awesome. Well, thank you very much, Dr. Darville, for coming to talk with us tonight and answering all of our awesome questions. Thank you, Cameron, as well, for giving your wonderful introduction talk. Kevin, is there any closing words you wanted to have?
Just to thank everyone for coming, and we'll be back with you all next week with a session on cardiovascular health. Thank you again, Dr. Darville and Cameron. This is really awesome. Great way to kick off 2022 Mini Med School.
Round of applause. Woo! Tell your friends about next week. Everybody come along.
Thanks again. We'll be back with you guys next week. Please email us at our Gmail account, uncminimedschool@gmail.com, if you have any questions about registration or any difficulties with Zoom or anything else. Thanks so much.
Thank you, guys.
Yeah, you're welcome. There's a question in the chat about when we'll get the recordings up, and we'll try to get that up by tomorrow evening.